Okay, so thank you very much. Welcome once again in the second session, second part of that lecture. This is the page where I have the reference seq information, and this is the nucleic acid sequence of the first transcript, and I'm clicking it. Remember the name, that is very important. The code is very important because this code is specific for that transcript only. कहीं पर चले जाएंगे, ensemble में चले जाएंगे, तो वो NCBI के इसी कोड को याद रखेगा. कंसेंसस CDNA सीक्वेंस पे चले जाएंगे, वो NCBI के इसी कोड को पहचानता होगा. ये बायोइन्फॉर्मेटिक टैग्स हैं which the system can recognize. So I am clicking here in the nucleotide sequence of the messenger RNA to reach that page. That page also contains the sequence information, protein sequence information, and certain other features as well. Remember, the term features is important, and it is used a lot in bioinformatics. The feature means you allot a name to particular part of the sequence. So, ye exon hai. So naming that portion as an exon is like giving a feature to that part. Like this is an 5' UTR region, so giving that portion a name is called featuring that name. And you should know how to add and remove features as well, not on the NCBI, but in your own hands. So can you start with the understanding? Here is the length, 465 base pair, length of that thing, the species name, and look at the code, the word. This is the same code which I have clicked seconds, a moment before. 0.3, this one is the same code. And then there are other related information once again, if you want to note down those information and the references and the papers from where they get that data. I will go down to show you the products and the gene sequences. And look at that. Here are the features and here are the locations and descriptions. Features. So what that mean? What is the total length of the insulin product? 465 base pair. So it says the gene starts from 1. Look at it. It starts from 1 and ends where? At the 465 base pair. That makes sense. Everything is a gene. And where is that everything? The everything is here. Look at that. At the end of the page, there is the sequence of that gene. It was divided into fragments. Each set contains 10 base pairs for easier counting. So this is like the base pair number 1. And this one should be the 10, 11, 20, 30, 40, 50, and this one should be the 60. So guess what? This one will be the 61st. This one will be the 421st, 430, 440, 450, 460, 461, 462, 3, 4, and 5. So 465 total base pair from here to here exists in insulin. That exactly was the feature is telling you here, 1 to 465 base pair. Then it's an exon from 1 to 42. Then there is another exon from 43 to 246. Or these are features. And then there is CDS. You know what is CDS? It's a more important feature here. The most important feature is the CDS. CDS is consensus cDNA sequence. This is the portion which is used to create proteins. It should start from where? It should start from the start codon. It should end where? It should end at the stop codon. Let's try it. NCBI allows us to click that sequence. If we click on that CDS, it will show and highlight that portion for us as well. So easy. So, abhi maine pehle bataya aapko, it should start from where? The start codon. That means the first 3 base pairs should be ATG. Look at that. This is A, this is T, this is G. Can you tell me the number from where it is starting? It is starting from the base pair number 60. 60, 61, and 62. These three base pair are the start codon. And let's go back to see the CDS once again. The CDS starts from where? It starts from 60 and it ends where? It ends at 392 base pair. So that means at the 392 base pair there should be a stop codon. Let's see on that sequence. Here, look at that, TAG, and TAG is a stop codon. So it starts with a start codon, it ends on a stop codon, and that portion is called as consensus cDNA sequence. That means this is the portion everybody believes is utilized to create or to synthesize the protein of insulin. That is the portion. Now, whatever the sequence present above that thing, ye sequence, whatever the sequence present above that portion, ye kaun hai? Shabash. Answer. Text or voice. Voice में करेंगे मुझे text box भी खोलना पड़ेगा, but that's fine. Whatever the sequence present above the start codon, so it is the promoter by one student. Yum, 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 yum. It's the untranslated region of that gene. Remember, the promoter is present before, even before that thing. It's called the untranslated region or 5' UTR. Shabash. Whatever the sequence present after the stop codon from here to here, that is the untranslated region present after the stop codon, because the gene sequence contains these three fragments. The central portion used for protein synthesis, the first portion untranslated region, very important for translation. The last is 3' untranslated region, very important for the translation, but the translation is done using the sequence present in the CDS region. And these symbols, the double hash and these things, these are bioinformatic signs. You cannot miss any of them. If you are using other languages program in the future, abhi aap Allah kare, inshallah karenge, then all these things matter. They are actually telling something to the computer. Okay, yahan par khatam ho gaya hai, yahan par start ho gaya hai. Their placements, these, I'm talking about these double hash and something like these things and the numbers, they are all have means to be there. In sabke matlab hai. You can't miss any of these things in the future. Now the next important thing जो हमें करनी है वो क्या करनी है, how we can keep the record of that DNA sequence. देखो अगर मुझे cloning करना हो, if we want to do cloning of the insulin, the most common thing we are doing in our labs, so I should know the sequence of my product and then my gene, and then I need to know the sequence of the plasmid as well. What if the resultant sequence, if I clone it, what will be the resultant sequence when I restrict that plasmid? So we should know all those things, but the first step, how to keep that sequence in your own computer. You are not like supposed to go every time to the NCBI and clicking that many times and end it up like this. You need to keep that sequence in your way, which suits you. And nobody else in the world will be able to decipher those things, because this is your research, this will be your PhD, this will be your publication. So how you can keep that thing? The most easiest way the people are using, they copy that stuff. Most easy, chill. और एक और बात बता दूं, protein sequence. Sorry, नहीं बताया मैंने. The translation product. Look at the translation product here. It starts from M. It should start from M. The first codon, the methionine. So this is the protein sequence. You can copy and save it in for your thesis as well. Okay. Okay. So I have an intruder. Just make a magic for them so that they can work better. Let's have a magic. Okay. This is a fairy giving you the blessing so you can understand this thing. Thank you very much. Okay. So this is the protein sequence. You should keep that sequence as well. For what? Because if somebody asks you, your professor is so furious, oh, to kitne base pair, kitne kilodalton ki protein banegi, or just let me know how many amino acids are there. You should have the specific record of your work with you always. You should be ready. You should be the one who know everything about your product, about your research. But फिलहाल अभी हम इस sequence की बात करते हैं. How people are keeping those sequences, they just copy and paste that sequence, which I am doing right now, and then you can have a dedicated folder for that thing. For example, I am creating a dedicated folder for this purpose, my name, date, and whatever. And remember, try to be as much specific as you are. ये हम इतनी गलती करते हैं, ये आप सब लोग मेरी बात से इत्तेफाक करेंगे. We just keep saving the things. कल को कर लूंगा, परसों को कर लूंगा. Pictures, you are saving the pictures, and then after that it will be very difficult for you to go back and browse the things and get the things back. This is study, this is research. You are not supposed to be lazy. Whenever you are saving, this is the best time to record and save the things in a specific way. So for example, I would love to write like Biot 42A class demonstration insulin, something like that. Okay, I should add date as well, but the computer is putting the date in the folder for me. You know, the folder is saved with the date and all these things. So how I will going to save it? I will save it the most common way. I will create a text file, insulin, nucleic acid, sequences. I'm not going to spend that much time on the naming, but you should write the name, you should write the date, and any information you can put in the name. So kal ko parson ko, it will not be trouble for you कि आप किस sequence को देखने हैं. I am putting that sequence here. Control V. The sequence is there with me. So now it is saved with me. Control S, it is saved. So that is the way most of the people believe they should keep the DNA nucleic acid sequences. There's another way, a better way. What that is? Microsoft Word, insulin, nucleic acid sequence with some additional information, blah, 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 whatever you want to write. The date, your name, your code name. Everybody used to have like certain code names. You can put your code name there. So I am opening the Microsoft Word. I'm asking Microsoft Word to keep my DNA sequence as well. Now here is the trouble which you should know. In the meanwhile, I'm telling you when it is here. So here, so you can place and write the title as well. You should nucleic acid sequences, gene name, whatever, blah, blah, blah things. And I'm pasting that sequence here as well. Okay. It is pasted in a highlighted version, but forget it. That's fine. It can help me. Thank you very much for the NCBI. It gives me in a highlighted version. So the trouble in keeping those sequences in this way is that it is very less efficient, because I am just giving you a most simplest of the example. I am thankful to the student जिसने insulin recommend किया था. Otherwise, we might be ended up in a sequence of like 4,000 base pairs, 5,000 base pairs, and all these things. But that is very small protein. So the Microsoft Word and the text file, they are word processors. They are word processors. They think whatever you are putting in, these are words, alphabets, words, paragraphs. They think about the numbers in a different way. You think about the words A, C, T, G as alphabets. They have no idea their DNA sequences. They have no idea there exist codons. They have no idea they should be translated into amino acids. So all these things the Microsoft Word cannot understand because it's a language word processor. If you change or delete one particular thing here, you will not be able to understand what actually happened to the gene. So this is the most inefficient way of keeping the DNA records. What are the efficient ways? There are lots of efficient ways out there. If you have money, then it will be much easier, because you can purchase wonderful softwares. But if you don't have money, you don't like to spend money, you should look around for the free softwares which can help you to keep the nucleic acid sequences and manipulate and edit it for you. There are lots of free software available out there. You can try, and you might be ended up in something better than this. But I am recommending you right now as a starter, Serial Cloner. This is the freely available software, but at the same time, it is giving us almost all the things which we required as a researcher. It will take you to this website. You can download it from here. And of course, you are more smarter than me. There are Windows version, Mac version, and there is an installer option there. Download options are there. So you should install like something which suits your computer system. You can download it as a zip folder. You can download the installer if you are using the Windows, and then you can install it. This is not one of the best. This is the free best. Okay. The one I remember I used to have like a software, $5,000 software in South Korea. They're rich people, for single user license for one person, for the use of one person at the time. But this is like free, so everybody can use. Of course, it does not give you the best of the best, but free is free. So when I install that software, I already installed it, already have it. It will give you that type of window. You can download and install the serial cloner 2.6.1 might be the latest version here. Okay. And now I will teach you how to keep. There are a lot of things we can do with this software, but as a starter, how to save and keep the file and how to make features in that specific file as well. Click on the file, make a new file, and then paste the same sequence here. Can you see I have pasted the same sequence? It's very small. It's very difficult. You need to have a very good eye. I'm really sorry about that thing. I can't, because of free version. So it's a little bit difficult to see things, I mean, more, in an easier way. So I'm reading it for you when you can see comfortably on your own computers as well. I have pasted the insulin sequence in the serial cloner file. Look at that. It tells me that it has total how many base pair? 400. Let me see. I am clicking Control A. Control A means selecting everything. When I select all the sequence, it gives me information here that whatever I am selecting, it starts from 1 base pair and it ends in the 465 base pair. The total length is 465 base pair. The DNA topology is linear. That means it's a linear sequence of nucleic acid. In case of plasmids, it is circular. Okay, you keep remember. The type, it is a DNA sequence. Okay, and these are the basic things you need to know. If I select certain other portion like this, if I'm selecting certain other portion like this, here I can see I am selecting from the base pair 53 to the base pair number 82, and the region I have selected is like 30 base pair length. So whenever you select certain portion, it will help you to know from where you are starting and from where you are ending. Remember, the word don't have any idea about all these things. It's just a way to keep the record of a sequence, that's all. Not the base pair numbers. And it is not taking it as a DNA sequence. This software is taking that sequence as a DNA sequence. I will tell you and explain other tabs as well. Just keep your patience, okay? It's not easy to know everything right away. Now, coming back to the NCBI page of insulin, which tells us that the CDS sequence starts from the base pair 60 and it ends at the base pair number 392. So since I am developing my own file, because I need to explain to my professor every now and then, I need to explain to share things with the other students as well. So I should have my own file with my own things. So I want to highlight that portion in that sequence. It's starting from where? I will type here. It's starting from the base pair 60. So I will ask the software to start it from 60. And then it should end where? Let me recheck again. It should end at the 392. Ask the software to end in 392. And then I'll push enter. It will highlight that sequence for me. So this is the sequence which we can call as consensus cDNA sequence. The software can search for us. It starts from where? You can see. It starts, oh my God, I have, मैंने entrance पे click किया तो I have lost. I cannot click on anything else at that time. Otherwise the information is gone. So Look at that. It is starting from ATG. It is difficult for you to see right away, but when you are doing the same thing on your computer, it will be easier for you to read the same things. Believe me, if you cannot see at that time, just believe me, it is starting at ATG and it is ending where? At TAG. TAG is the stop codon. And this portion is called as the consensus CDNA sequence. I want to feature that portion in my way. So this software allows you to feature it. How we can feature it? I will click, look at that, on that tab called feature. There is a tab called feature here. After selecting that portion which I want to name, I will click on the feature. Please remember the steps. After clicking on the feature, I will click on this plus sign, means add a feature. I will click on the plus sign to add a feature, and then I can name that feature in my way. I will name it consensus, whatever, consensus CDNA sequence or CDS, whatever. I can choose which color I want to put that region. I'm putting the red one, so it is should be more visible to you. That sequence, the information is telling me it starts from 60 base pair, ends at 392 base pair. I can add some additional description here of my own choice. I want to create a mutation at the base pair number 85 to 90. I want to delete those 5 or 6 base pair from 86 to 90 base pair. I'm just putting some random example. Whatever the things you want to write along with that sequence and feature, you can write here. Okay? I was taunted by my professor that I don't know what is CDS. So whatever the things you want to record, and then you can add some other things as well. Other things is a primer sequence, or it's an operon, or it's a binding sites, protein binding sites, or whatever. But right now I'm just leaving it as a miscellaneous feature. When I have finished that portion, that thing, I will go back to my original sequence. You can see the portion is highlighted in red. And when I click on that red portion, it will give me the name that you are, behold, you are in the consensus cDNA sequence region. It starts from ATG, you know that, it ends at TGA stop codon. I can beautify that thing further. I can add more features. Let me play with it. You should play these things. That I am selecting these 3 base pairs. ATG. I have selected it. Believe me, it's very small, but I have selected 3 base pair. You can see here, I have selected from 60 to 62. Let me name that portion. First step, clicking on the plus sign, adding more feature, giving it a name. I'm naming it start codon, and I'm giving it the beautiful color of green, so it is like a start codon. I'm not adding anything else. I will go back and see it is colored in a different way. If I click on this green thing, it is telling me the portion is in the consensus cDNA sequence and it is named as start codon. What about the stop codon? I can TAG, this is the stop codon. I select that portion, go to the feature, plus, and I name that feature as stop codon. Write whatever I want to write here. Choose the color. I am using the color blue this time. Go back and the color is different. So this is like the stop codon. What about the portion here above the start codon? That portion starts from 1 to base pair 59. आपने मुझे खुद बताया, what is that sequence, that portion? I will name it feature. I will add plus, then write the name. This is the 5' UTR region. That is a place where RNA pol 2 understands the transcription start site and start making the messenger RNA. 5' UTR region. I can write whatever blah 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 things I want to write here. I can choose the color. How about the pink? And then go back. It is colored as a 5' UTR region. You can keep on going. I mean, you can make things in your way easier for you to understand, to get the things, and to share it with the rest of the world. This portion feature plus 3' UTR. Once again, giving it a pink color. It's a UTR region, for example. Come back. So everything is colored. It's beautiful. Now, what more that sequence can do for you, that software can do for you? It cannot be done by the word processor. You need a software which understands the difference between the alphabets and DNA. How that software can help us to understand about the sequence we know. Remember, we placed a very small sequence. You can place like 5,000 base pair, 10,000 base pair sequence. You can do whatever. There is no limit in using that software. Next, I am going to click on the sequence map. Another tab I am showing you, the sequence map. When you click on the sequence map, it will show the same sequence. Oh my God, where is that? Here. Are you able to see my screen?
No, sir.
Okay, sorry. I think I have clicked at the wrong place. Sorry. Okay. When I click on the sequence map, it shows me, it takes me here. Okay. Lot of buttons are here. It will make you a lot of confusion for you. And I can tell you, I mean, bioinformatics sometimes help me a lot about the understanding of my molecular biology. Because when you see the things, it gives you a different impression. Can you see my screen now? Okay.
Yes, sir.
So there are a lot of things out there. You can change the size. Of course, we should, ताकि हम इसको बड़ा करके देख सकें. मैंने maximum 14 points कर दिए. And there are 3 translational frames here. Frame 1, frame 2, frame 3. You know what is translational frames? Every one single DNA sequence can generate 3 protein translational frames. And we don't know which one of them is actual or functional translational frame. I will show you which one. I can pick which one of them I want to see and which one of them I don't want to see. I recommend you to click here, restriction sites. You know, there are certain restriction sites present on our sequence. Cloning के लिए important होती हैं, दूसरे purpose के लिए भी. But right now, this is not required. So I want them, do not show restriction enzyme sites. So it is not showing me anything, any restriction enzyme. It's become very simpler, you can see. So this is the sequence once again in a different way. Every DNA sequence has a double stranded, so it has an opposite strand as well. Can you see there's a single sense strand? And then you can see the antisense strand at the same time. And look at the feature we have placed. We colored this sequence as red or as pink, 5' UTR. We named this one as a start codon. Then the red region is CDS. Then we name this portion blue one as a stop codon. And then there is a pink portion 3' UTR. So it is showing us the other sequence as well, the antisense or nonsense strand sequence as well with our features. Okay, out of these frame, which is the correct frame? The correct frame should install a methionine at the start codon, and it should install a stop codon at the end of the consensus cDNA sequence. So out of these 3 frames, these are the protein sequence in between. The protein sequence in between. I got a question. Of course, we can do XPACY. Why not? Why not? ये तो घर की बात है ना. It's in your own hand. One of the students asked me that why can we use XPACY. The answer is yes. There are hundreds of websites which can help you to translate. Okay. नो प्रॉब्लम। लेकिन ये हमारे घर में, हमारे सामने, हमारे हाथ में एक सॉफ्टवेयर जो हमारी मदद कर रहा है इसमें। ओके, so we have 3 frames out there. So which of the 3 frame, which one of them is the perfect frame out of it? I told you the perfect frame should be the one which we should install methionine using ATG here. This should be the perfect frame. The first one is not installing methionine here. The second one is not installing methionine here. The third one is installing the methionine here. So out of these 3 frames, the functional correct frame will be the frame number 3. So that means I don't need to see the frame number 1. I will uncheck this box. I don't need to see the frame number 2. I will uncheck that frame as well. And what I will be left with, the frame number 3. And that is the correct frame of the protein sequence. So what you are looking at right now, sense strand sequence labeled in your way, anti-sense strand sequence labeled in your way, and you can see the protein sequence as well, and there should be a methionine. One another way of confirming the sequence, the frame sequence, is that the correct frame should not have any stop codon before the termination of consensus cDNA sequence. If I believe that the first frame is the right frame, it is not starting with the methionine, so it is not the correct frame. And you can also see it is installing a stop codon in the middle. So that is incorrect. I can't use it. If the second frame is supposed to be, I'm putting the second frame now, is the right, it is incorrect because it is not installing the methionine here. It is incorrect because it carries a stop codon. It's not correct. What about the third one? It's a perfect translational frame for the insulin because it is installing the methionine here, and you can see there is no stop coming, no stop coming here, no stop coming here, no stop coming here unless we have the end of the CDS sequence here, the TAG, then we have the stop codon. So that is how we can confirm this sequence. So we not only keep the DNA sequence with us, but we are also keeping the record of the opposite strand and the protein sequence as well, along with that sequence. The Microsoft Word have no idea about all these things. When you put the sequences in Microsoft Word, it has no idea about all these things. If you want to search the sequence in Microsoft Word, every space will cause it trouble. These numbers will cause it trouble. Enter will cause it trouble to search a space, but these things will not cause any trouble here in that sequence. You can search it right away. How to search a specific sequence? Let's suppose I want to search these base pairs, like base pair G, C, 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 T, G, G, T, G, C. I will copy that sequence and I want to see where that sequence exists in insulin. All I need to do, I'll control F. Look at that, control F. When I click on the control F, it will give me an additional window somewhere here. You need to sometimes search or find it, sometimes hide itself. This is the control find option of that software. It gives you the searching option using nucleotide. You can search it using the protein sequence. You can search it using a restriction enzyme site. Wonderful. And as well, you can find it the open reading frames as well. But I am searching using with the help of a certain base pairs. So I am putting those base pairs here, and it will tell me that these base pair exist here. 271 base pair pe pade hain. If I click, it will take me to the place from 271 onwards to 280. So we can search any sequence. We can search the sequences which are present on the sense strand, and we can also search the sequences which are present on the other strand. The software will understand the sequences which are present on the opposite strand. Okay, from the internet you can convert those sequences very easily into sense strand into another strands. For example, if you simply write the reverse complement or Google anything, it's so easy. The platforms are there. You can always find options which can help you to convert a DNA sequence into its reverse complement form or only into reverse form. So for I am putting that sequence here. I will submit it. It will give me the product. This is the product. So this is a reverse complementary sequence. If I search that sequence in the Microsoft Word file, it will not give me any result. You know why? It will not give me any result because it says no matches, because that sequence belongs to antisense strand. That sequence does not belong to the sense strand. It's not here. If I search the same sequence, for let me start from Control F. If I search the same sequence here, it will show me the place that the sequence exists here. You know what I am searching? I am searching GCA. It is giving me GCC. I am searching something else. It is showing something else, because the sequence which I am searching is not present on the sense strand. It is present on the antisense strand. And that is highlighted here. Look at that, minus. So whatever the sequence I am searching is not present here, it is present on the opposite strand, and the software is telling me by putting it minus here. Okay, so that is how we can search the same things in a better way in this software. I will keep telling you more and more about these things, and I request you people to please practice with some of 4 or 5 genes out there. Okay, and I would love to thank all of you once again to be here with me in this class. Please practice 4 or 5 genes and try to put them onto the serial cloner, and then try to create those files and save those files as well. I did not show you how to save it, but that's very simple. You just need to click on the file and then you need to save, and then write the name of your choice, and that is how you will be able to save that file very easily. Okay, so I thank all of you once again to be here.